Hello and welcome to today's webinar, Catching Aggregation Early. My name is Dave Dolak. I'm the Sales and Marketing Director at Brookhaven Instruments. Today we have our speaker, Daniel Seeman. Daniel's Chief Scientist at Brookhaven Instruments, where he has served as Scientific Advisor and Subject Matter Expert since 2019. His work focuses on light scattering and particle characterization methods across a range of applications, from biopharmaceuticals to colloids, and advanced materials, including the use of dynamic light scattering to assess the stability of proteins and aggregation detection. Prior to Brookhaven Instruments, Daniel was postdoc scholar at the NIST Center for Neutron Research, working within the macromolecular and microstructural group and the NSOFT consortium. He holds a PhD in chemistry from the University of UMass Amherst and a BS from Stony Brook University. Just a couple ground rules before we get started. Please post questions in the comment section in the chat, and we will keep track of those and have a Q&A at the end. So without further delay, I introduce Daniel, my colleague and friend. Take it away, Daniel.
All right, Dave. Thank you for that introduction. So I'm just presentation. Should be. Go. All right. So, see, so everybody's here. Good. So the topic of today's webinar is basically detecting aggregation of monoclonal antibodies using dynamic light scattering, specifically under low pH stress conditions. So as Dave said, I'm Daniel Seeman, and I'm going to be presenting today. So a little bit about Brookhaven. So we have been in business for over 4 decades. We primarily manufacture and design light scattering instrumentation. We have other types of particle characterization equipment, but our focus since the beginning has been light scattering. We have instruments in essentially virtually every major university, national labs, and industrial labs. So we've been around for a long time. We have a dedicated team of experts here to help you out with whatever you might need. So move this thing out of the way so I can see the slides. All right, so just a quick overview of the talk before I sort of launch into everything. If you take away 4 things from this presentation, one is that it is virtually impossible to avoid low pH stress in the purification of monoclonal antibodies. Essentially between protein elution and viral inactivation, antibodies are basically always going to be exposed to a low pH environment for at least some amount of time. We're going to talk a little bit about formulations. We're going to talk a little bit about analytical development, and in general, we're just going to talk a lot about light scattering and how you can use it to measure monoclonal antibodies. Specifically, we're going to talk about resolving monomer versus aggregate, and we're going to talk a little bit about data interpretation, and hopefully you're going to take away some practical advice for performing light scattering experiments that you can use in a industrial environment. So a little bit about how we're doing this. So we have a few different DLS products. The majority of the data for this presentation was collected on the BI200SM, which is our multi-angle static and dynamic light scattering goniometer. So basic principles of DLS, you're basically measuring translational diffusion. You plug everything into Stokes-Einstein. You hopefully know the temperature and viscosity. You measure diffusion coefficient. You do a little bit of algebra, and you calculate a diameter. Then the goniometer allows you to do this at multiple angles, so you have an arm that can basically rotate around the cylinder. So every DLS has at least basically these 3 components. So you need a laser, a sample holder of some kind, and a detector, and then typically a dedicated signal processing device, in this case a correlator, specifically our turbo core, but just a digital autocorrelator card of some kind to do signal processing. Specifically for the goniometer, we have a 40 milliwatt red laser, which is our standard laser. Specifically for this experiment, a 640 nanometer, well, for all the data collected today, a 640 nanometer programmable red diode laser with a source power of uh, maximum power of 40 milliwatts. A sample cell that sits in the middle of an index matching vat to deal with reflections, detector optics, um, and which are basically used to focus the uh, emitter of the sample cell, and an APD, which is an avalanche for the diode, which is what produces the actual signal that gets fed into the correlator and then it rotates around the sample. So why even perform a DLS measurement at multiple angles? So there's an implicit assumption anytime you use a DLS to measure particle size, which is that you're measuring a translational diffusion coefficient. If you're measuring a translational diffusion coefficient, then if you produce this plot called a gamma q squared, you should get a linear dependence between the decay rate that comes from the correlation function and q squared, which comes from the scattering angle. And every single angle should give you the same effective diameter, and the slope should effectively give you the diffusion coefficient. But it's simply large molecules diffuse slowly, small molecules diffuse very quickly, and DLS is basically just measuring the speed of those dynamics and then making a few geometric assumptions and calculating the diffusion coefficient from them. There is no right or wrong angle to perform a DLS measurement on. On a benchtop DLS, most commonly you'll have a high back scatter angle, a low forward scatter angle, and then sort of the default on most qubit-based systems is a 90-degree or right angle measurement. On a goniometer, you can basically sample any of the intermediate angles as well. So a little bit about the software we use to collect this. So we have a software suite called Particle Explorer, which contains dynamic and static light scattering functionality. Today, we're just going to talk about DLS. This is basically a combination of data acquisition software and data analysis software. Our DLS module allows you to automate a number of different variables. The most common forms of automation will include angle dependence, temperature dependence, time dependence. You can program these all systematically, and you can loop over a number of different control variables. And so you can basically use Particle Explorer to drive an angle dependent experiment. And then you can either use the built-in data analysis modules, or you can export the raw data if you'd like to write your own. So why are we talking about monoclonal antibodies today? So we're talking about a near $500 billion market. Basically, all the major next generation blockbuster drugs for decades have been monoclonal antibody based. They're basically magic bullets for autoimmune disease. They're used for treatment of autoimmune diseases. They're used in vaccine research. They're used as components in immunoassays. They're very expensive to manufacture, very expensive to formulate. They're formulated at extremely high concentrations, have to be completely resistant to aggregation, and QC failures are incredibly expensive. So again, a little bit about monoclonal antibodies. They are large, flexible proteins made up of multiple subunits. The sort of cartoon version is that you have these antigen binding domains at the very end, and then typically for pharmaceutical mAbs, you'll have a human IgG scaffold that you make very conservative changes to. Since they're injectables, you obviously want something that isn't going to produce an immune response, so you want to stay as close as possible to the sort of canonical human IgG scaffold, and then you change basically just the sticky bits at the end. But as people have observed for decades, small changes to the antigen binding domain can result in major changes to the solution behavior of monoclonal antibodies. And also in contrast to like traditional compact small globular proteins, mAbs are big and they're dynamic. And they're also held together by that series of disulfide bonds. And so even a small amount of unfolding can result in potentially irreversible aggregation. And then obviously pH, ionic strength, and temperature can all affect this. And then as we said earlier, low pH is unavoidable when you're purifying monoclonal antibodies. So we really have to know, you really should know how your specific mAb responds to low pH conditions. So obviously protein A chromatography is essential first step in the purification of any MAB, especially at industrial scale. It's an affinity resin. It's a very expensive affinity resin, and you basically concentrate your protein, and then once you've washed away all the impurities, you elute with a low pH buffer. Frequently, this is coupled with a viral inactivation step, so you can avoid having multiple low pH exposures. So viral inactivation, because much of the time when these things are made in cell culture, you're using viral transfection systems, and so obviously you want to make sure there's no live virus in the final product. So not only do you have to acidify monoclonal antibodies as part of the production process, you have to hold them at a low pH for a substantial amount of time. And unfortunately, a lot of characterization work happens downstream. So the person characterizing a formulated MAB may not necessarily be in the same building as the may not necessarily be in the same building as the person who's actually doing the initial capture and activation steps. And so you have to make sure that there's some feedback. Another possibility would be bringing DLS further back in this procedure. So you may have a person either in QC or formulation who has more time to dive into SOP development, so they can develop a procedure for characterizing monoclonal antibodies. And if they identify a small amount of aggregate, unfortunately, they don't really have any input until after you've gone through this whole process. So you could potentially take the same SOP and then just move it further upstream, including potentially sampling earlier in the process, or you're doing probably live process monitoring. So back to the specific experiment we're talking about here today. So we're starting off with a formulated pharmaceutical grade monoclonal antibody, molecular weight of around 160 kilodaltons. It's formulated in dilute citrate buffer, and then a 1 to 50 dilution brings it to its final concentration for DLS. If we measure immediately after acidification, we see a small amount of aggregation. So you see an effective diameter above the expected 12 nanometers and a high polydispersity index. Just a little bit of settings. We're doing this at 90 degrees to start with. But we see, obviously, an effective diameter larger than 12 nanometers tells us that we're not seeing entirely monomer. If we want to get more detailed information, we employ a multimodal size distribution with two standard canonical multi-mode algorithms, including the most commercial DLS software, are NNLS, or non-negative least squares, and content. In this case, we're looking at NNLS results, and you can see that whereas there is a well-defined and clearly identified monomer peak, we also see potentially two different size aggregate peaks, although it is difficult from the single measurement of a potentially rapidly aggregating sample to necessarily say too much about this peak. So we can be fairly confident in monomer. We can tell that there is some aggregate present, but it is difficult to say much about it. We can also see that the majority of the signal on an intensity basis is coming from our aggregate, despite the fact that we're probably, by number, almost entirely monomer, with a very small amount of potentially submicron or larger aggregate, potentially one single aggregate passing in front of the laser transiently during experiment. And because it's intensity weighting, which is basically just saying that, in this case, 65% of the signal came from this large peak. That does not mean that 65% of your sample is contained within this approximately 4 micron peak. So we started off talking about an effective diameter. What is that? So the term Z average shows up a lot in the literature. Z average radius, Z average diameter. That's basically referring to what we call the effective hydrodynamic diameter, or the cumulance diameter. Cumulance is a very simple model that basically has two fittable parameters that come out. One is basically a mean effective diameter, and the other is a polydispersity index. And these two things together give you an idea of what the average size is, and basically the how much that sort of deviates from the assumption made in the model. the cumulance model is not capable of producing multiple peaks. The model makes the assumption that there's a single population with some width or some breadth that's measurable. So a very low poly dispersity would be effectively indistinguishable from an undispersed sample. An intermediate poly between 0.1 and 0.2 most likely would describe a single population that happens to be highly polydispersed. And with a PDI above 0.2, that's really where you shouldn't be using cumulants anymore, as it no longer is representative of the sample that you're trying to characterize, and that's where it's usually more appropriate to switch to multimodal distributions if you want to get anything quantitative in an experiment. As we saw before, we're able to reliably pick out a monomer peak. We're not really able to reliably characterize the aggregate peak other than simply knowing it's there. So as I mentioned before, there's a lot of assumptions implicit in a one angle TLS measurement. Since we have a goniometer, we're going to do an angle sweep. So we're plotting the cumulance diameter versus Q, or versus Q squared, which is a function of the scattering angle. So small q's are low scattering angles. So this end we have forward scatter, on this end we have back scatter. We notice that if we try to do a best fit line to our gamma q squared, it deviates pretty substantially. That implies that the cumulance model is not doing a good job of actually describing the population present. If you happen to be sitting in certain points along this best fit line at certain scattering angles, you might get a more or less representative diameter, but that would depend a lot on basically where you are. If we take the same results and we do a little bit of machine learning and a little bit of clustering, so we take our NNLS results, we do a little bit of clustering just to divide these up into two different populations, because peak assignments for multimodal distributions can potentially be a little bit twitchy. We group all of the fast modes together, all the slow modes together. We get a best fit line that is does a pretty good job of describing the monomer, and then potentially a few different best fit lines describing the aggregate. So again, it can be a little bit difficult to say much about the aggregate other than the fact that it's present in the sample. We look at the individual correlation functions, we can kind of see why. So we know from light scattering, on an intensity basis, larger particles will scatter more light substantially. We also know that as we go to lower scattering angles or forward scatter angles, we greatly amplify the contributions from these larger species, as you can see from these correlation functions. So you see relatively at backscatter, you see a relatively clean autocorrelation function with a single observable decay. We get closer to 90, you see a small secondary decay start to appear at essentially lower decay rates, or a second slower moving species present. But if we go all the way to 30 degrees, our lowest forward scatter angle in this experiment, we can see a pretty large contribution due to our aggregated species. So our fast mode or fast dynamics on the left, slow dynamics on the right. As we shift scattering angle, we not only shift this correlation function, but we also potentially increase the contribution from the larger species. So we can see if our goal is to simply detect dust with the highest sensitivity possible, or detect aggregate more commonly with the highest sensitivity possible, we want to go to the lowest scattering angle. If we want to measure the size of a monoclonal antibody monomer, then we want to go to backscatter. We want to measure that with the highest accuracy. And if we want to be able to detect both with reasonable accuracy, we probably want to be sitting around 90 degrees. So we look at some of the time-dependent data. So looking at a single time point post-acidification gives us some information. But if we acidify the sample and then collect data continuously over the course of 2 days, we can see that initially you start to form large aggregates. You almost immediately start seeing multiple peaks on an intensity basis. Here we're looking at, so basically 48 hours worth of data was batch exported from Particle Explorer and then fed externally with basically Pythonic, like NNLS and content implementations, and just doing that to visualize the data. but you can basically see two things here. One, which is that at initial times, you see a lot of aggregate formation. And then the sample starts to become more uniform as you form larger species and they settle out. You can also see that N and LS and content frequently agree, but there can occasionally be small discrepancies just due to idiosyncrasies in the algorithms. So in any individual DLS experiment, your raw data is basically a single autocorrelation function, and then a count rate history, which is basically the instantaneous sample count rate taken every 1 second during the measurement. We also have something we call a dust rejection algorithm, which basically, in a given experiment, will selectively omit dust-contaminated data from the running average of the correlation function. So for the time-dependent data that we're about to look at, we essentially have a time series that is made up of a number of individual correlation functions and count rate histories. So we look at all the data together. So time 0, rapid acidification by addition of a small amount of nitric acid. What we're seeing here in black is the average count rate for an individual measurement. And what we're seeing in gray is the max count rate observed in each individual count rate history. Again, a little bit of clustering. We pick all of those out, plot them, look at them as a function of time, and we can see if we look at the mean count rate, there's a lot of aggregate formation in the first 10 hours of the experiment. As the aggregates become larger, they drop out, and so the mean count rate starts to drop. You get these transient spikes where you form aggregate, and then it settles out, you form more aggregate. As we saw earlier, you'll typically start to see like a shoulder in the correlation function when you form larger species. So one thing that we're able to do is set a threshold for max count rate, take all this data, and essentially fish out good measurements and bad measurements. So bad measurements where there is a discrepancy above a certain percent between the max count rate and the baseline count rate. We can see in both cases, you can typically see multiple peaks in the good measurement. So good measurement does not necessarily mean unaggregated sample. It just means a good quality correlation function, and that we're under experimental conditions, we're able to produce good DLS data. So you don't want large amounts of material drifting in and out of the beam path during an experiment. So if all you have is precipitated or aggregated protein, you're not going to be able to produce fittable DLS data, which is really the condition we're looking at on the right. And on the left, you can see over the course of time, we start off with monomer at the earlier times. We form a little bit of aggregate, but you can always see the monomer peak, even if it's not necessarily the only peak or the dominant peak. Zoom in on these, so you can see 13 hours in, we see a lot of monomer. We consistently see aggregate peaks appearing and disappearing. We do occasionally see smaller peaks, which could be potentially degradation products, or they could just be experimental noise. If we look at the essentially poor quality data, the sample is so aggregated at these specific time points that there's just really no way to fit it sensibly. All right, so if we summarize everything, aggregated, we've basically covered why explain why we're interested in the aggregation of monoclonal antibodies under low pH stress, because there's no way to avoid it when you're producing monoclonals in a pharmaceutical environment, because you basically have to use protein A for capture, and you have to do viral inactivation at low pH. So these are not avoidable steps. By the time it gets to QC or formulations, there's it's usually a little bit too late to fix anything, but you can take what you learn one end of that pipeline and potentially use it to develop a better test that can be done earlier on in the process. So hopefully this provides some practical advice for using dynamic light scattering for studying protein aggregation. And we talked a lot about how you can resolve monomer and aggregate populations. It's difficult to quantify an aggregate. It's very easy to detect an aggregate. And just because there's aggregated protein present does not mean that there's no monomer present. It's pretty much most of this is generalizable to any type of DLS platform. Specifically in this case, we used our research goniometer, the 200 SM, to make all these measurements. However, every one of these experiments, with the exception of the continuous multi-angle experiments, could be reproduced on our NanoBrook, which is our fixed angle or benchtop instrument. And certainly a hybrid approach could be using a goniometer to develop an SOP and then implementing it on a NanoBrook so that you can, or any benchtop system, so that you can test earlier on in the pipeline. And these techniques are all complementary. Any questions?
Thank you, Daniel. Appreciate the presentation. So I would like to thank everybody for their time today. Thank you, Dr. Seeman, and have a good weekend no matter when and where you are. Take care. Thank you.
Thank you.